Hey, welcome to the Bioinformatics Chat. This is the second episode in a row when we will be talking about Markov models. This time we'll be talking about hidden Markov models, and we'll be putting them to good use. We'll be using them for genotyping based on long-read sequencing. And one of the authors of this algorithm is today with me, Trevor Pesout. Welcome to the podcast.
Hey, thanks, Roman. I'm happy to be on here.
Trevor, tell us briefly about yourself and your background.
I have done a lot of my work in computer science. I started the PhD program at UC Santa Cruz and found my way into a bioinformatics lab and have since changed majors to join the bioinformatics department. I have done a lot of work in method development, specifically for long reads, under the advice of my advisor, Benedict Payton. I'm a fourth year student now. Probably have 2 or 3 more years left of the program.
Right, and that's at University of California, Santa Cruz.
Yeah, that's correct.
Cool. So one of the concepts that we'll be dealing with today is long-read sequencing. So give us a brief introduction. I think it came up a couple of times in this podcast, but what is that? And why is it very different from the normal sequencing, the next-generation sequencing we're used to?
Yeah. So I think the biggest difference that I usually talk about is the length and the quality of long reads. So next generation or second generation sequencing produces relatively short but very high quality reads. So the nucleotide sequence you get, you're pretty sure that all of them are correct. For long reads, the reads are of a much lower quality, so you'll get base called errors, maybe 1 in 10 or 1 in 100, and they oftentimes will have sort of large gaps or large insertions in the reads that just aren't present in the sample, and this is especially so for nanopore reads. So the PacBio, Pacific Biosciences, and Oxford Nanopore technologies are the two big long read sequencers. I've got a lot more experience with nanopore reads. Nanopore reads are not quite as accurate as PacBio, but they're generally longer. I think they've got a lot more potential for development.
We should also mention a piece of recent news that PacBio got acquired by Illumina.
Yeah, that's right. And I hope that means that PacBio is going to keep moving forward. We'll see how that develops, but I'm sort of hedging my bets on Nanopore being the long-read sequencer of the future for a lot of my work.
Okay. And what kind of instrument do you use? Do you use MinION?
Yeah, so the lab at UC Santa Cruz has had a long-standing relationship with Oxford Nanopore. One of the people who developed the idea met back in 2004, 2006, works at Santa Cruz. And so we've had a long history of work with ONT in the development of it. So we had one of the first MinION sequencers, and that's still around, but their latest sort of large-scale sequencing platform of PromethION has also been in use at Santa Cruz for a year, maybe 2 years at this point. We do a lot of work with Miten Jain, who is in the department, who's our resident nanopore expert.
Did you get a chance to play with his instruments yourself, or are you confined to the dry lab?
I'm much more of a dry lab guy. I have gone into the server room where they keep it. It is surprisingly small to see. It's maybe 2 feet by 3 feet. It's pretty impressive how much sequencing data can come out of a relatively small little machine.
For sure, yeah. Of course, you can do a lot of things with long-read sequencing. One of the recurring themes, so one of the main applications of long-read sequencing is assembly, de novo assembly, when you need to resolve long repeats and connect the contigs. But you apply long reads to something maybe a bit less conventional, and that is genotyping. That is figuring out if you have an individual and you sequence their genome and you try to figure out which alleles they have at like various SNPs locations, right? Is that a common thing to do? Are there any precedents? Are there any existing algorithms to do genotyping from long reads?
Yeah, there have been a few papers that have come out in the recent past trying to do this, but one of the biggest problems is that the quality of long reads is just so poor that it's really difficult to use the existing next generation algorithms and applications in order to do genotyping and variant calling. With short reads, it's you do an alignment and you can essentially count up the nucleotides at each reference locus and call it a het variant or a homozygous variant. But because the reads are so lossy, it's not really possible to do that with long reads. We, during the development of this project, we tried running, for example, GATK's haplotype caller on long reads, and we couldn't get anything. We ran Freebase on it, and it called a variant about every 5 nucleotides. So these methods really aren't set up for it.
Right. So it's a bit paradoxical because when doing genotyping, you actually care the most. You don't care about the big picture. You care the most about this very tiny point variations. And therefore, it seems a very bad fit to use the technology that gives you a good big picture, but very lousy sort of single nucleotide resolution. So how did this idea come about to do genotyping with long-read sequencing, and what are the benefits of long-read sequencing in this context?
Yeah, so it came about, my advisor, Benedict Payton, and another researcher, Tobias Marshall, who works out of Max Planck in Germany, came up with the idea together. And we'll go into it, but the loose idea is that we should be able to leverage the fact that these long reads will span multiple heterozygous sites, where we know that if a read comes from one of these maternal or paternal haplotypes, the presence of adjacent heterozygous sites enables us to divide the reads into mom and dad chromosomes, into the maternal and paternal ones. So that we'll go into it, but that's sort of the crux of the method. But like you said, why would we want to use long reads for this? And the reasons are because there's a lot of areas in the genome that are difficult to call with short reads. So especially in repetitive or duplicated regions, short reads have a difficult time mapping uniquely. So, for example, centromeres and telomeres have satellite DNA, and it's very long stretches, megabases of the same small motifs repeated over and over again. And variation in there is hard to tease out because reads can map exactly to many areas of those regions. Similarly, in duplicated regions, so you can look back at gene duplication events, transposons such as lines and signs. These cause the same sequence to appear multiple times in the human genome, and in a lot of mammals too. And so, again, if you have a read that has come from one of these regions, it's hard to figure out exactly where it fits into which one of these lines or signs it does. So the benefit of long reads is that they can span entirely these genomic features. Sometimes they'll be anchored on one side of it and extend into it. So, for example, if you were trying to see if there was some variation in the portion of the centromere of a chromosome, you could. It's much easier to find a long read that uniquely maps to one of the non-centromeric or pericentromeric regions, but then will extend into the centromere. And if you can use that to get visibility into the centromere, you can start to call variants in there. You can start to understand the variation that exists in there. Some of the nanopore runs that we've been getting out of the Prometheon sequencer with Matein's help produce reads that are megabases in length. Maybe not 2 megabases, but over 1 megabase, which means that we can really get a deep vision into some of these hard to reach areas of the genome.
Right. So on the one hand, you can genotype these repetitive areas that you normally don't have access to. And also as a free bonus, you get this phasing information. So you know which haplotypes there are in this sample, so which SNPs are clustered together on the same chromosome, essentially.
Exactly. Doing the variant calling with long reads sort of as a byproduct of this method means that we also get out information, like you said, about haplotypes. So finding variants that are all inherited together also has clinical applications. For example, if you had a variant in some coding region that is associated with another variant in the promoter for it, that variation is going to behave differently from the case where there's something that changes the promoter for a region, but there's a deleterious variant in the other haplotype. So knowing which variants go together is also an important benefit of using this method.
Yeah, that's a good example. But okay, so we decided that there are certain benefits to doing genotyping with long-read sequencing, but still we have to overcome this problem that long reads are very noisy. They have very high percent of errors. So the figure usually cited for PacBio was 15%. Do you know what the percent error rate is typical in your experiments?
I think that the technology is improving. I think that 15% is maybe a little more errors than we actually see with PacBio. One of the things, though, to understand is that with short reads, the errors are pretty much randomly distributed. So, and especially with the presence of quality scores, you can use them to say, well, the read shows a miscalled base here, and we can be sure because it's a low quality read. With long reads, the errors are systemically patterned. So, for example, with nanopore reads, the base caller has a hard time correctly calling homopolymers, so multiple of the same nucleotides in a row. So, in addition to this effect of having miscalled bases, having insertions and deletions where you wouldn't expect, you also get these areas where the genome has, 10 A's in a row, a poly A tail or something like this, And the nanopore sequencer will consistently miscall those, which means that the error profile it's different, and it's
So maybe a simple percentage doesn't really tell you much, because when we say like X%, or like 1% for Illumina, we mean that if you look at a specific base called, then it has this probability of being incorrect. But if like a whole segment is removed or inserted, it's not as easy to reduce to a single percentage, perhaps.
Yeah. The point I was trying to get at was that with short reads, with enough sequencing, those errors will be sort of removed. If you have enough read depth, you can sort of just by the law of large numbers tease out what's an error and what is a true variation. With long reads, you can sequence 10, 15, 30x, 60x, and you'll still get the same methodological errors in the reads. You'll still not be able to tell how many A's there are in a row, because it's always going to consistently miscall this area.
I think it's different for PacBio versus Nanopore, because I know Gene Myers, he talks a lot about how the errors in PacBio are random, and so he makes exactly that point, that with PacBio, if you have sufficient depth, then you can get perfect assembly. And perhaps with Nanopore is different. Yeah,
you're right. That's a good point. I've like I said, spent a lot of my time focusing on Nanopore, and you're right. That is the error model for Nanopore, but PacBio is a different beast. I think you had originally asked what are the error rates in reads, and I don't think I actually answered it, but for Nanopore, I think it's closer to 20% of the reads are off in some way.
How do you define what the 20% means? Is it like if you align the read to the genome, then like 20% will be mismatches or something?
You'll get about 20% or 80% identity. So that includes insertions, or it doesn't include insertions. It doesn't include deletions in there.
Oh, it does not. So 20% is just if there is a base there and you call the base, then 20% of time they will be different. But that doesn't include all the possible deletions and insertions, which adds to the error profile.
Yeah.
Right. And so, that makes it very tough to do genotyping. So how do you work around that very salient issue?
So, like I said, the idea that we're trying to leverage is the fact that you can use the heterozygosity of the sample to bipartition reads into the two haplotypes. And we've set up this hidden Markov model that will go through the alignment and find areas where we think het sites are, or where there are potentially errors in the sequencing, and statistically determine what is the most likely partitioning of reads. And then once you have that information, the genotyping process becomes much easier. You're able to just sort of look at the haplotypes. You can call a consensus on them and then use those to determine variant sites.
So the idea is that haplotypes are sort of less noisy, right? Once you separate the reads into the reads coming from, chromosome A versus chromosome B, meaning the two homologous chromosomes, then it's much easier to call variants if you know that all your reads come from the same source.
Exactly. It enables us to look at, we know that all those reads have come from exactly the same sequence. So if we do have the perfect bipartitioning, which is not necessarily the case, but if we had it, we would be able to look at all the variation in those reads and classify it as errors or true variants.
Right. But my understanding is that although it may be convenient to explain the algorithm as these two phases, so first we do phasing, we do, we separate reads into haplotypes, and then we do variant calling. But I think in your model, it actually is, all part of the same model. It's all done simultaneously. Is that correct?
Correct. We do it in the model we set up simultaneously does the phasing, the haplotyping, and the variant calling.
Right. And what allows you to do that is the hidden Markov model. And this may be a good time to introduce this concept. And then we'll see how it actually helps us to get where we want.
Cool. Yeah, that sounds good. So I think that in order to understand what a hidden Markov model is, it's important to take a step back and look at what a Markov model is. So a Markov model is a statistical method that you can use to model different systems. So it's got a lot of applications. You can model weather patterns and populations of species and gambling outcomes and things like this. And the idea is that it's a stochastic process, and you model it with a state machine. So you have some set of states that the system can be in, and there are likelihoods of changing from one state to another at different time points. And associated with each state is an emission. So I think that's sort of the key structure of the model. You've got different states and you've got different emissions at each state.
So emission means something that you actually observe as opposed to states in the hidden Markov model. So the word hidden comes from the fact that you don't observe the states. You know that there are some states and there is something jumping from one state to another state. You don't actually get to observe in which state the system is in, but you observe some kind of projection, some kind of manifestation of that state. And there is no deterministic one-to-one mapping between states and observations, but there is a probabilistic mapping. So like some states, in some states, you're more likely to observe some one thing, and in other states, you're more likely to observe some other thing.
Yep, that is exactly it. It's much more difficult, I think, to conceptualize what a hidden Markov model is because you don't know what state it is. All you get to see are those emissions, the direct observations, and you have to use those to infer what the underlying states are. Exactly. I think a good example of this that I got from the textbook that I learned how to do hidden Markov models from. It's called Bioinformatics Algorithms, an Active Learning Approach by Pesner and Campo, is playing dice with somebody who has a loaded die. So, for example, if you're playing dice and somebody has a die that is loaded so that it's much more likely to roll a 6, and also a fair die where all the sides are equally likely, and this person has some chance to swap dice. So you know they're going to, if they have a die, if they're using the loaded die, they'll be much more likely to keep using that loaded die. But at some point, they may switch dice. And if you're trying to determine whether the die they're using is loaded or fair, based just on what the outcomes are, what the rolls are, I think that's a good conceptual example of how a hidden Markov model works.
That's a good example, yeah. And now if you have to map this problem of genotyping and phasing onto this mathematical abstraction of hidden Markov model, how do you do that? What are the hidden states and what are the observations in your genotyping model?
Yeah, so we've decided that a hidden Markov model is a good way to describe this problem of haplotyping because you've got a well understood biological process that gives you pretty natural states. So you know that each read is either going to come from the maternal haplotype or the paternal one. So each read is of one of two states. And then the emissions are the reads themselves. So given you're in the state, I'm examining my maternal haplotype, I have some chance to see a read emerge from there, and that read should exhibit the variation that exists in that maternal haplotype. In our method, the states themselves are actually modeled in terms of all the different partitionings of the reads. So I think the simple version is just to think of it as each read is mom or dad. But in actuality, our method looks at each read aligned to a reference locus and then enumerates all the combinations of states, of maternal and paternal states. So, for example, if I have two reads aligned to one place, at that position, our model has four states. They're either both maternal, both paternal, mom and dad, or dad and mom.
Right. And to clarify, the individual states correspond to individual loci on the chromosome. And so moving from one state to the next state corresponds just shifting by one base to the right along the chromosome.
Exactly. And then set up within these different states, we have this idea of compatibility. We know that it is biologically impossible to get a read that starts half on the maternal chromosome and then ends on the paternal chromosome. You're not ever going to see that. So set up in our state machine is this enforcing that if a read was in a maternal haplotype at one state, the next state and all states after that, it will continue to be in the maternal haplotype.
Right. So this is how you arrange your hidden states that from a given hidden state, it is possible to get to some other states, meaning states at the next nucleotide, specifically to the states that are compatible with the current one, that do not contradict logically the current partitioning of reads into the two classes. And so from each hidden state, it is possible to get to some states, to the compatible states, and it is impossible to get to any other state. So this is how you arrange your probabilities. And then among those possible transitions, do you assume that they are all equally probable?
Yeah, we model it as in at adjacent reference loci. We don't really distinguish between, the likelihood of one thing over another in terms of those transitions. We can look at the reads themselves and use those emissions to determine the likelihood that one partitioning is more probable than another, but the transitioning from one state to another is all equally likely.
Right. So if you are in a hidden state, then your probability to transition to another hidden state, to the next hidden state, is either 1 over n, where n is the number of outgoing edges, or 0 if that next state is unreachable, if that's not compatible with the current one.
Exactly.
So this is one component of the hidden Markov model. That's the hidden part, the hidden states that you actually want to get some insight into, right? You do want to know how those reads are partitioned and what the genotypes are. We should mention the genotype is also, maybe we mentioned it, the genotype is also part of those hidden states.
Yeah. So for each one of those hidden bipartition states, we also have states within them representing the likelihood of, or the possibility of, the different genotypes. It is sort of an added complication on top of the model, but the way the hidden Markov model works, it's relatively easy to add states on top of that. You can sort of think of the bipartitioning as a larger state that you can be in, and then internal to that, you have multiple states based off of what those possible genotypes are. And then, of course, the aligned nucleotides at that locus do much more to inform what those are, what the genotypes are.
Yeah, so you put enough information into the hidden states so that you can meaningfully judge the probabilities of those emissions. So if you know how the reads are partitioned between the two chromosomes, and you know which chromosome has which allele, then you can say, okay, this is what I observe from all the reads aligned to this locus. This is my observation. And then you can judge how likely this is to be observed given the partitioning of the reads and the genotypes.
Yeah, exactly. You got it.
And at this point, do you embed any particular error model? Because this is where you have to judge, right? Is this just a sequencing error, or is my assignment incorrect? So do you assume any specific error model for the reads?
Yes. Yeah, that's a great question. We actually have two slightly separate error models that we include in our calculation for this determination. So the first one is sort of based in biology. It's a biological substitution model. And the rough idea is that we would expect there to be a variant every 1,000 nucleotides. About 1 out of every 1,000 bases is mutated with respect to the reference. And then furthermore, on top of that, we expect that transitions are a little more likely than transversions. So this is what nucleotides will mutate into other ones. So we have that error model. And then additionally, we have an error model that accounts for the way that the sequencers will miscall bases. So, like we said, Nanopore and PacBio have different error profiles. So we have the same biological substitution model for both read technologies, but Nanopore and PacBio have different read error models. And we did training to determine what those are. I think that it's an imperfect system, and especially because as the Nanopore base callers change, those read error models also change. So that's something that we kind of have to continually analyze. Baked into the MarginPhase program is a method that will go through and generate an error model. It essentially performs the bipartitioning once, looks at the expected haplotypes, and then calculates how far off from those haplotypes the reads themselves are, and uses that to generate a more accurate error model. So you can do training of the model with the application itself. It's sort of a expectation maximization method.
And the error model that is computed, is it just the probability of an error, or how does the model itself, the error model for sequencing, how does it look like? Like how many parameters does it have?
It's a full substitution matrix. So it says that given the read was an adenine, what are the different likelihoods of the actual underlying base being an adenine, a cytosine, and so on. So for every nucleotide character, it gives a likelihood of what is the true nucleotide based off of this observation. And that helps a lot in the determination of what is the genotype, is that error model.
Okay, so that accounts for base miscallings, but how do you model the insertions and deletions? Do you have to model that?
Yes, that's a great question. So we did a lot of experimentation with treating deletions and insertions as special characters. And what we found was that it caused all sorts of errors to accrue up in the output. We found that if we, for example, treated deletions as its own character, as a gap character, that the quality of our results dropped drastically. Again, because there's so much insertion and deletion in the data. So at some point we decided that this was a problem that we were going to tackle in a different way down the line, and that our method would just focus on doing snip calling. So for both WhatsApp and MarginPhase, the two implementations we wrote, we decided to disregard indel variants. In terms of the model, what we treated this as, we treated deletions as missing data. So instead of saying, oh, I observed no aligned nucleotide here, I'm going to draw some conclusion of that. We just treated it as, hey, there's no alignment here. This read doesn't have any information in this location. And we're not going to use that to make inferences about underlying genotype.
So by now, we just defined our mathematical model. We defined, here's our data, the reads, the long reads, and these are our hidden states, and we sort of defined, given the states, how likely it is that we will observe this kind of data. But that is, of course, not yet an answer. We have to get from the model to the actual assignment of states. We want to know at this locus which one is the most likely state there. And so how do you get from the model to the answer?
Yeah, so when we are dealing with hidden Markov models, there's really two different methods that we use to analyze these. And they both run off of the idea that we're going to use dynamic programming to interpret the observations with respect to the hidden states. So if you're unfamiliar with it, dynamic programming is a method where you consider all possible states that you could be in, and you set up a matrix that has the states as one axis and the observations as the other axis. And because the likelihood of being in a particular state at a particular time is entirely dependent on the observation and the likelihoods of being in the previous states, you can make a single pass through this matrix and get an idea of all the different likelihoods at the different states. So it's just a single pass to go through all this information. So when we're doing this calculation, there's two different algorithms that we use for understanding hidden Markov models. The first one is the Viterbi algorithm. So in this case, we run through the dynamic programming matrix once, and we're trying to find the most likely path through these. So, for example, you can run it through and then take the most likely state at the end of execution. And then from there, you go backwards and you look at what's the most likely state that that we were in previously. And then given that state, what was the most likely previous state? So you do this back trace through there to find a single path through this state machine. And that path then represents a sequence of states that you're in. This is one method. For us, we actually used what we call the forward-backward algorithm, and it's a little more complicated than the Viterbi algorithm. What we do for forward-backward is we run through the dynamic programming matrix once in the forward direction, and then we essentially invert the model. So instead of going from previous observations to the current observation, we go from the last observation backwards. And once you've done this, that backwards pass gives you a slightly different set of states. Once you have both of these dynamic programming matrices, at each time position or for each observation, you can find the exact distribution of state likelihoods. It's a little counterintuitive, but you don't quite get that information by just doing a forward path, because you don't consider from the forward direction all the possibilities generated or controlled by the observations later than that, which is why you need this backward path. So once you've done these two, you can combine the likelihoods of all the states at a single point in time, and you can find the most likely state that you were in at any particular time. So for our method, we run forward, backward, and then at each individual time point, we can see what's the most likely bipartitioning of the haplotypes. And then we can pick the most likely one of those and output that as our final result. I know that was a lot of maybe programming or algorithms there, but I think that it's actually quite simple once you wrap your head around it.
Yeah, the way so I think you gave like a more computer science explanation, because the algorithm itself, like what's happening inside the machine, is just a dynamic programming. But the way I would explain that from a statistical point of view is that to figure out the distribution, so you have a bunch of hidden states, and it helps if you imagine them as being like very linear as nucleotides on the chromosome. So it's a linear chain of states. And if you want to focus your attention on just one of these states, then statistically what you want to do is you want to what's called marginalize, or to sum up all the irrelevant states. You want to run through all possible assignments of values to other states and sum them up, and that will give you the exact distribution at this particular state. And that's essentially what the forward-backward algorithm does. So the forward pass is essentially summing up everything to the left of the current nucleotide, and the backward pass summing up everything to the right. And once you sum it all up, that will give you the distribution of the current locus.
Yeah, that's exactly right. And you're right. I do tend to think about things in a computer science or algorithmic sense. But yes, that marginalization is where we got the name for the MarginPhase program that we wrote.
Interesting. I didn't realize that.
Mm-hmm.
That's funny. Also, I don't know if you're aware, but it's a pretty cool thing. I learned about this recently from Leo Pachter's book. There is a deep connection between the Viterbi algorithm and the forward-backward, or actually like the forward part of the forward-backward algorithm, and that is essentially the same algorithm. So, they're both dynamic programming algorithms, but they are like really the same algorithm if you consider like two different algebras. So like if you run the Viterbi algorithm but change the algebra from multiplication and addition to addition and taking the minimum, what's called the tropical algebra, then it's really the same algorithm, and it just blew my mind. It's a very beautiful fact.
Yeah, that's really cool.
So the forward-backward algorithm is what tells you sort of the most likely assignment of states, or actually for each state, the distribution and the most likely genotype at that point, and also the partitioning at that point. But there is one issue, and the issue is that your Markov model, your hidden Markov model, is ginormous. And that's because you try to consider all the possible partitions at each point, and there are 2 to the power of n, where n is the sequencing depth at that point, and of course you need a high sequencing depth in order to reduce the noisiness of the reads. So you do need big depth, but that will lead to a huge Markov model, and you have to deal with that. So how do you deal with it?
Yeah, that's a great question. So in order to handle this case, because yes, as you said, the number of possible states increases exponentially. To start out, we take a maximum of 64 aligned reads at any particular position. So if you have a read depth of 100, we essentially randomly discard reads until we get down to a maximum of 64 depths. Which
and what is, by the way, a typical sequencing depth in your experiments?
For long reads, we found that we did a lot of our experimentation on some public PacBio and Nanopore data. So for PacBio, there was a Mount Sinai sequencing run that came out, I think, 3 or 4 years ago. It's available on the NHI and CPI website. And we found that had an average read depth of about, I think it was 45, something like that. And that statistic is after we pruned out all reads that had a map quality of less than 30. So that means that all the reads that we kept had an error rate of 1 in 1,000 in terms of being aligned correctly. That's kind of different from an average read quality, but we were pretty sure of the alignments. So for the PacBio stuff, about an average depth of 45. How large your BAMs are for a normal sequencing run really depends on how much sequencing you want to do. So it's a little hard to say about what's the average amount, but we found during analysis of the data that at about a read depth of 20, our precision and recall started to converge at close to its max. Before that, you get a lot of variation in the quality of the calls.
But you did have some data sets with more than 64x coverage, or otherwise you wouldn't have come up with the heuristics, right?
Mostly we picked that because in C, it's written in C, and we can get a 64-bit integer to mask the bipartitioning. I think that the reason for 64 is that it was well above what we should need, and it fit easily into a into a common data type.
I see. Yeah.
So that bit mask is what we use to describe what we think the bipartitioning are, or to enumerate all the different bipartitionings. And I don't think I mentioned, but the Nanopore BAM was in the middle 30s. I think our average depth was about 35. And that came from Matane Jane's Nature paper published in, I think, 2017.
So you never actually bumped into the ceiling of 64?
That's not entirely true. The alignment, the read depth varied across chromosomes. We did a pretty thorough alignment or analysis of the alignment. And there are regions where by chance or by, some random factor, we would have a very high read depth.
Ah, I
So
see.
there are areas where we did have to prune reads down. But
Right. So,
yeah.
so you is it at each locus you choose your 64 reads max?
The way we set it up is by doing a single pass through the BAM, and we greedily pick reads, and we set them up into these things we call profile sequences. So we take reads that are non-overlapping and set them all up into a single path. So we take the first read that we get, and then as soon as that read's alignment terminates, we take the next read that starts that is aligned after that end position, and then we iteratively go through and generate these profile sequences until we have either reached a maximum at our location or all reads in the BAM are accounted for.
Mm-hmm.
So it's not really at every reference locus we pick a set of reads. It's that we use as many reads to generate a depth of 1, and then we use as many reads as we can to generate a depth of 2, and so on.
Right, right. Okay, so we were talking about how you deal with the exponential explosion of the number of partitionings.
Yeah, exactly. So we use an algorithmic tool. It's sort of a divide and conquer method. And so what we do is we take these very complicated set of states and we divide it into half. So we recursively take the set of profile sequences and we drop them down until we just have 2 sequences together. Then we run our algorithm on this forward backward, and we can use that to find the most likely partitionings of these small sets of reads. So we essentially prune out unlikely states. Then, having done this for all pairs of reads, we combine those pairs into a set of 4 reads. And then once we have 4 reads, we can run the algorithm on it again, only on the more likely states, and then use those likelihoods to prune down other additional unlikely states. Having done this multiple times as we iterate back up through the stack, it ends up taking longer to compute, but the state space that we use never gets that full exponential growth because we've removed a lot of the states as we go down through this stack and back up again.
That's pretty cool.
Yeah, it's a cool method. The method still takes a lot of memory. Part of the issue with this is that it still does have a high memory footprint, but it's not that exponential, thousands of gigabytes of memory. It's sort of on the order of tens of gigs, we found.
And does it depend only on the sequencing depth, or does it also scale with the chromosome length?
It does scale with the chromosome length also. So when we ran MarginPhase, we took and developed a pipeline to enable us to chunk up the input chromosomes, divide it into pieces, and then run MarginPhase on sort of bite-sized chunks of it, and then use that information to sort of stitch together the output VCF. We used a workflow system called Toil, which was also developed at UC Santa Cruz. It's a Python framework that relies heavily on Docker images, which enables you to have some measure of security that your generic workflow is going to be run in the same way each time. With genomic workflows, you find that somebody updates a new version and all of a sudden your expectations have changed in terms of what the output data should look like. So it relies on these static images and enables you to, for example, run the workflow on the Amazon spot market. So you can still use the cloud infrastructure, but it lets you back up your workflow and checkpoint it at various states. so that you can utilize the cheaper spot market, which ends up costing somewhere between a half and a quarter of the cost generally than
Right.
using
So for
standard
people
instances.
who are not aware how this works, is essentially you can borrow the time on the machines that are idle, so you don't get a guaranteed machine. But if some machine is idle at Amazon, you can utilize that a bit of time.
Exactly.
For a fraction of the price.
The benefit is that it's much cheaper, but the cost is that as soon as Amazon finds somebody who actually wants to pay full price for that hardware, they'll shut you down without warning. So being able to checkpoint your work is a critical part of being able to use the spot market effectively. But we found that it really is an effective method to do high-scale computing.
So a natural level of parallelism here is chromosomes, because they're mostly independent. But is there more parallelism? Like, can you process different parts of the same chromosome in parallel on different machines?
Exactly. That's also one of the benefits of being able to chunk it up. So the workflow that we ran is fully configurable, so you can choose any chunk size that you want. But we ended up doing, I think, 3 megabase chunks at a time, which enables us to do essentially 1,000 of these at once, given a 3 billion or 3 gigabase genome, which means that this method makes it very parallelizable.
So say I wanted to run this on my laptop. I think I have 16 gig of RAM, so the RAM should be enough. But in terms of processing time, is there any hope that I will live to see it finish?
So the MarginPhase implementation is at a whole genome scale probably going to be difficult to run on your laptop. But the paper that we put out actually included 2 different implementations of this method. So the other method was developed by Tobias Marshall, like I said, who's at Max Planck, and his grad student, Jana Ebler. And they built it as an extension onto WhatsHap. WhatsApp is a existing tool to do haplotyping. And their implementation is efficient enough that it can run on a laptop. Part of the benefit of, or part of the reason that they enable you to do that is because they require that you have existing variant sites beforehand. So it's a little easier for them to, or for the WhatsApp implementation, to run and to consider fewer states. The MarginPhase implementation does everything sort of de novo. So you end up considering a lot more states, which means that it takes up a lot more footprint. So if you were trying to run this application or this method on a laptop, I would recommend looking at the WhatsApp implementation instead of the MarginPhase one that I worked on directly.
That's interesting. So running your algorithm, one version could run on a laptop, the other one probably not. But you also mentioned that the alternative variant colors also didn't run. And what were the issues there? Is that they were taking too long to run on long reads? Or so you said, I think, so Freebase found a lot of variants, so probably its error model was off for long reads. But what were the issues that you ran into?
Yeah, I encourage people to explore different options, but we certainly found that all the short read variant callers we used either didn't complete, couldn't even start. In the case of GATK's haplotype caller, it just depends on too much of the short read infrastructure, for example, quality scores and things like that. So we couldn't get that to run. And yeah, Freebase produced pretty awful results in terms of its precision, and took prohibitively long when we ran it. I'm not an expert on it, so it might be that I missed some sort of, default setting or something like that. But I think you'll have a hard time trying to run those short read callers on long read data.
And did you compare your algorithm to the long read callers, the existing ones?
We did. There is a preprint out called, I'm not sure if it's Clairvoyant or Clairvoyante. And they use a neural net to do base calling. So I'm not sure exactly what their method is, but there's been a lot of progress in the field doing convolutional neural nets into relational neural nets to do image calling. Or to do base calling, variant calling, excuse me. These methods will look at essentially a pileup of the reads and can use sort of existing image processing frameworks to try to tease out what the variation is. Some of the existing methods will do local realignment of the reads. So, for example, if you have indels, it'll try multiple different alignments around these loci to try to determine these. The problem with using one of these methods is that you don't get the haplotyping information unless you can account for the long range information in these reads. You can't really infer haplotypes from it, which is part of what we were trying to do with this method. There's another paper put out by a guy out of Hong Kong, Guo, who has a method where they look at long reads. I believe that they find a read that best matches the other reads, and then tries to bipartition the reads based off of similarity to those exemplar reads. But we weren't able to get it to run on actual data. They did a lot of their analysis on simulation data. So as far as we know, we couldn't find another method of getting both haplotypes and variants out of long read data.
Yeah, now that you mentioned it, I remember it was a big piece of news maybe a couple of years ago when, wasn't it like Google or some other big player that came up with this algorithm based on like reducing it to an image? Like people, I'm not sure if everyone caught it. It's like literally an image. It's not some kind of bioinformatic concept. It's like literally a picture that they draw this pile up picture, and then they run some kind of picture algorithms on that. I found it very funny at the time, but did you have any success running that?
Yes. So you're right. Google did do this. The application is called Deep Variant, I think. And the way they set it up is you can sort of imagine a picture of your cat or something as a collection of pixels and color depths. And the way they encode genomic information into this image is with different channels. So I don't fully understand it, but I think a good analogy is they'll take all the red pixels and they'll say, we're going to encode the nucleotide information here using this set of using this part of the image, and we'll encode the support track, whether or not these nucleotides show support for a particular variant in this track, and we'll put quality score using the blue pixels or something like this. So when you look at it, from what I've seen, it looks kind of like a single plaid t-shirt pile up. It's pretty incredible what they can do with it. And using those methods enables them to use a lot of pre-trained knowledge on images. So I don't know if we want to go too far into it, but there's a lot of existing training that they've used neural nets to do to, for example, pick out shapes and things. So if you want to look at a picture and determine, oh, is this a dog or a cat? You start out by taking the shape of the things that are in there, and you can compare colors and all these different features of the data set. But using those, it enables you to expand upon all this existing training. In terms of our method, we obviously are using a statistical method, which I tend to like a little more than relying on neural nets because it's understandable. Or, I mean, there's some complicated math there, but at each point, all of the calculations you have some sort of meaning. Neural nets tend to be a bit of a black box. They will produce good results a lot of the time, but you don't really know what's going on under the hood unless you're a very, very expert in the field. What we think is that in addition to these, different tracks that they put in these images, that if we could, for example, do a processing pipeline on data, and even if we don't call it variants, if we can divide the reads into haplotypes, encoding haplotype information into the images might provide a piece of context that can help these image-based variant callers, which do work very well. So we think that our method has a home in these workflows, even if we don't directly do image processing or variant calling using those methods.
Yeah, I think we try to do a good job explaining the model and the algorithm. And is there anything else that you'd like to talk about?
I don't think I have anything else to say. I think that using long reads is going to have a home in a lot of clinical settings in the near future. I especially think that the nanopore sequencing shows a lot of promise. They're still improving the pores that they use and the base colors that they use to mitigate a lot of the errors they currently show. One of the other good benefits of nanopore data, which I haven't mentioned today, is that they can do methylation calling, or that's starting to show this promise. And I think that once that methylation calling happens, there's going to be a big explosion of new work that's being done using that in both a clinical and just sort of method development sense. I'm personally really excited to get that working because I think that the presence of allele-specific methylation, so methylation that happens differently on the maternal and paternal chromosome, is going to enable this method to work better because we'll have more sites for differentiation and should hopefully be able to produce methylation variant calling, which is something that doesn't really exist and isn't well understood. So I'm really excited for the future of nanopore sequencing and the applications that it's going to have down the line.
So that's a really interesting point. So when you analyze just the genome sequencing, then you rely on the fact that the DNA is everywhere the same, right? So you can, because you don't sequence single cells, you collect a sample and you have many examples, many copies of the same chromosomes, and this is how you get your sequencing depth. But in the case of methylation, you cannot know that it's necessarily the same across different cells. So I'm not sure how this is going to work.
Yeah, I think it's not a very well understood part of the genome. Currently methylation detection is very hard and expensive to do. I know that there have been some studies that show that at specific locations, they see evidence of allele-specific methylation. Chromosome X inactivation in females is one of the easiest examples. But, we're not really sure if methylation exists randomly across other areas of the genome, whether it'll be that at specific sites we will always see methylation or not. So I think it'll be difficult to piece together evidence of methylation that happens consistently in areas, whether it's on one haplotype or both, and where methylation exists randomly. But I think that a hidden Markov model is something that should be able to describe that interplay. Adding in states to represent the methylated nucleotides, states to represent whether the sequence is being methylated in an allele-specific way or whether it's being methylated randomly. But I think that this method should enable us to tease that out once the data is there. So I think that's an exciting prospect.
Yeah, definitely. And I just wanted to say that I really enjoyed the way you applied the hidden Markov model here. I haven't seen anything like this before, so an application of hidden Markov model to genotyping, and I thought the model is really elegant. It's not something you obviously see. Like, if you consider how to solve this genotyping problem, then perhaps hidden Markov model is not the first thing that comes to mind. But once you really dig into it, it really starts to make sense.
Cool. I appreciate it. I agree. I think that it is a cool sort of, maybe not intuitive application, but I think that in practice it works out really well.